Good evening, everybody. First of all, I want to thank you for your presence and having the time to go over a wonderful story about an innovative diagnostic tool. I would like to present Professor Bruce Patterson. He received his MD at Northwestern University of School of Medicine, followed by a residency in pathology. He also began investigating cellular reservoir of HIV using molecular and in situ cell-based technology. He also was present in a group that worked on the HIV virus, and he published in Science in 1993 about this work. And Rolling Stones on a Discovery Channel, Dr. Patterson has authored over 100 manuscripts and books. Also now he serves as the CEO of incellDx, as a founder of incellDx too. And he has more than 100 patents for his work and under the incellDx program. Thank you, Dr. Bruce, for being here.
Thank you, Marcel, and it's great to be here in Lebanon. Number one, with Benta, our great partners here in Lebanon, who are helping to bring this innovative technology to both Lebanon and the Middle East. But I'm especially happy to be amongst colleagues in the pathology world, so we can talk about all the things pathologic. It's rare that you get to convene a whole group of pathologists in a room just to have a lively discussion about what we face on a day-to-day basis. What I'm going to talk about today is single-cell technology that we developed probably close to 15 years ago. Before anyone was talking about single-cell technology, we had developed a technology platform. We developed a single-cell technology where we were able to do comprehensive, qualitative, simultaneous, and multiplex detection of proteins, messenger RNA, DNA, and morphology, all at the same time in intact cells in suspension. And because we could do in situ hybridization in suspension with simultaneous protein detection on cell surface, we could actually combine protein and mRNA and do it by instruments like flow cytometry, or even newer instruments like CTC machines. When we did this in the mid to late 90s, we were able to actually quantify the number of HIV-infected cells in HIV-infected patients and then follow them after therapy. And basically what we did, we took the same strategy and applied that to cancer and detected both proteins and cells related to cancer, and we're able to both say what therapy they'll respond to, but more importantly, we were able to follow the destruction of those cancer cells following therapy. This is basically our technology. As I'll show you, we've developed a way to make single-cell suspensions using a FDA-approved device of ours. We're able to stain the surface of the cells with a multitude of antibodies, up to 20 now. We poke holes in the cells without destroying the antibody labeling. That's key. To do in situ hybridization without proteases, we hybridize mRNAs in a multitude of colors, and then we can come back with intercalating dyes and actually do cell cycle analysis and proliferation, all in intact cells, without ever destroying the cell. And we've maintained the cell morphology, so we can run this through a flow cytometer and you won't see any difference from fresh cells. Unlike some in situ hybridization, it causes cells to swell or change their morphology. We've been able to maintain that with a very specialized fixative. One of the first things we did, though, is we created a platform, a pre-analytical solution to every sample source. This is something we started at Stanford. We were trying to figure out a way that how we could take any sample in the world and prepare it in such a way that we could do any test available to us. And what we did is we created a line of products later on when I developed the company, where we could take fine needle aspiration, blood, or urine, and put it in a reagent called incellMax. incellMax is unique in that it is a mild, non-cross-linking fixative. It preserves antigens in their native conformation. It allows us to do in situ hybridization without proteases because it doesn't bind proteins to nucleic acids like the cross-linking fixatives do, and allows us to do DNA analysis with CVs less than 4%. So it's really a fantastic fixative. We're now using it also as a cytology fixative and as a transport media because RNA is stable for up to 2 weeks at room temperature, and proteins are stable up to 3 months at room temperature. So it's a very unique reagent that's approved both in the US and in Europe. If we have a fresh frozen tissue or core biopsy, we use a device called incellPrep, which is FDA approved now, to create single cell suspensions non-enzymatically. So we can take tissue biopsies, core biopsies, make a single cell suspension. And so the way we address the molecular world in the United States, when people come up to us and say, well, you need fresh tissue, you're against the standard of care. What I say is, no, we're not. How do you do lymphomas, right? I'll pose that to you. How do you do lymphomas? In the CAP protocol that's in the United States, you take a small piece, you push it through a mesh, and you do flow cytometry, right? All I'm saying is for every solid tumor, we should be doing that. We now have a device that will make that single cell suspension from a small 4 millimeter punch biopsy. And then the rest of the tissue could be submitted into, for processing into a block, okay? The last is, what do we do with FFPE samples, the holy grail in pathology in the molecular world? Well, we have a new kit called incellReveal where we can create single cell suspensions out of FFPE blocks and actually run them by flow cytometry. Okay? So what's the goal? Our goal is to get single cells in solution. Why is that important? Well, we can do flow cytometry. We can put these cells on a microfluidic CTC platform. We can do microscopy and cytology, and we can do PCR and NGS, all from the same sample. And the fact is, we can do that without it ever seeing an aldehyde. And again, that is significant advantages down in this area of this curve. I just wanted to show you our incellReveal because I think it's pretty unique. Look at these beautiful single cell suspensions out of a 2 millimeter punch or a 60 micron scroll. This was a real challenge for us. Took us 2 years to develop. And this is flow cytometry, actually on cells from an FFPE block. I don't know if you've ever seen this before. We actually stain for a variety of different markers, variety of different tissues, looking at various staining patterns. It's very unique, except we're faced with the same struggles that we are when we look at IHC. We still have to do antigen retrieval or use antibodies that are made specifically for formalin-fixed tissue. So what we've done is we've created this very unique product line, which we're just now introducing into Lebanon and the Middle East. We have Oncotect IO head and neck, which consists of HPV E67 messenger RNA and PD-L1 at the same time. The PD-L1 drugs have just been approved for head and neck in the United States. They're used extensively. HPV for head and neck, even though it's probably 30%, maybe 40%, is a prognostic marker for head and neck cancer. We can do it all at the same time on cells recovered using a swab of the lesion. Our Oncotect IO lung is also on the market. Consists of PD-L1 quantification, TIL quantification, and aneuploidy and proliferation using DNA cell cycle dye. Aneuploidy is still the most powerful prognostic marker in many cancers, especially lung and breast. Our breast diagnostic is unique in that we can do ER/PR, HER2 antibody, and FISH at the same time. So I know the standard of care is to do HER2 immunohistochemistry. If it's equivocal reflex to FISH, we do it all at the same time in a 4-hour assay by flow cytometry. We can also add in ER/PR, PD-L1, TIL quantification, and cell cycle all at the same time. Bladder, PD-L1, CTL quantification, aneuploidy, and pancytokeratin to define the transitional epithelium. I'll show you more on this later. We have a bioinformatics model that's allowed us to achieve 98% sensitivity for bladder cancer detection out of urine. And our Oncotech 3DX HPV, which has been on the market for the longest period of time, for about 6 years, is E67 messenger RNA quantification in individual cells and cell cycle looking at proliferation. So we look at 2 steps in cervical carcinogenesis: oncogene overexpression and cell cycle dysregulation and proliferation that results from it. We do this all, again, on high parameter flow cytometry. The Cytoflex is now here in Beirut, in Lebanon, at Benta and other labs now. They have a smaller version that has 3 lasers and 12 colors. This is 6 lasers, 30 colors by flow cytometry. There's a company called Fluidigm who likes to go out and talk about their ability to do, 20 and 30 color staining with their heavy metal labeled antibodies. But Fluidigm can't do cell cycle. And doing cancer analysis without cell cycle, as I'll show you in a minute, is like swimming without water. So the first thing I'm going to talk about is cervical cancer quickly. What we know, obviously, is you get infected. High-risk DNA actually just tells you you're infected. We wanted to do more than that. We wanted to be able to stage disease. We know during integration the L1 gene is knocked out, L1 gene being the target of the Roche assay, so there's some curious effects there. And the E2 gene is knocked out, which is a negative regulator of E67. E67 gets tremendously upregulated following integration, and because our assay is quantitative, Hologic, GenPro, Aptima is only qualitative. Other E67 assays for HPV are only qualitative. They don't tell you that you're actually overexpressing the oncogene like we do in our assay. And so we can look at the activity of E6, the activity of E7. This is from Harold Zerhausen back in the early 2000s, which he published in Nature Reviews. There's 3 steps that happen: oncogene overexpression, knockout of tumor suppressor genes, cell cycle dysregulation that lead to what? Proliferation and progression. We measure this with cell cycle dye, we measure this by in situ hybridization. So 2 biomarkers for cervical cancer diagnosis that allows us to say, are you a SIN1 or are you a SIN2, SIN3, or nothing at all, you're just infected. This was a very interesting paper that came out that shook the HPV world. What it did is it did a split sample analysis between 4 FDA-approved tests. HydroCapture 2 by QIAGEN, Roche Cobas, Aptima by Hologic, and Claret, which is popular in Europe. And the scary thing from this study was the concordance between all of these assays in a split sample study was 29%. Everyone talks about HPV DNA for screening. The problem is it depends on the test you're using. Each test had both false negatives and false positives relative to the other tests. And we're talking about 3 to 5 year screening intervals for women when the tests, depending on what you use, have false negatives. I wouldn't send my family there. So this is a big problem in HPV that nobody likes to talk about. In fact, at Urigen this year, you know what Hologic did? They put this diagram up and they eliminated this with Photoshop, the purple.
Wow.
Can you believe that? They didn't want anyone to know that. But I live by this diagram. So I went over and I said, why'd you eliminate this population? Well, it wasn't important to us. We just wanted to show them. No, it is important. Okay? It's extremely important because these are screening assays and there's 29% concordance depending on what you use. Okay, so for HPV, we want to incorporate actual biology. Cervical cancer is not an infection, but requires it. And understanding the dynamics of gene expression, cell cycle, and morphology is absolutely critical when we're evaluating the cytology. What we developed a few years ago is what we call the slideless PAP. We were able to take cervical cytology and put it in an instrument that did both essentially flow cytometry and morphology in cells in suspension at the same time. It had a high-speed camera that took pictures of the cells that went through a laser, and it did all the other things that flow cytometry normally does. So here's normal squamous epithelium. I don't have to explain to this group, very low nuclear cytoplasmic ratio, very small nuclei, very homogeneous nuclei. We can actually quantify the nuclear cytoplasmic ratio than trying to estimate what it is on the slide. Nuclear area, DNA content, it was diploid, and then we can look at E67 as well in this channel right here, which you won't see on normals, but you'll see here on an H-cell, you see these very odd morphologies. My brilliant cytology mentor from Northwestern in the 1980s used to call these tadpole cells. I don't know if everyone's used the term in this room. It was Denise DeFrias, Marlucci Bibo were the ones, we used to call them the brilliant Brazilians, who taught us all cytology. And these were called tadpole cells. They were tumor cells. They're overexpressing E67. Look at the heterogeneity in nuclear area, which of course we can measure if you put in an instrument like this. And we can measure the high nuclear cytoplasmic ratio. And we can look at aneuploidy by looking at, or proliferation by post-G0G1 and aneuploidy by DNA, all at the same time. Now we were trying to push the fact that we could use this instrument to do away with having to look at every single slide. However, it would have cost about $50 million to get that through the FDA. So we went the flow cytometry route where we could do DNA index and E67 at the same time. For anyone who's looked at flow cytometry and they look at dot plots, you usually look at antibody versus antibody, right? This is the first dot plot that's messenger RNA versus DNA. That's where we are today. And this technology is sitting right down the road in the laboratory. And so what we were able to figure out is that E67 gets overexpressed in the G0, G1 population of the cell cycle in low grades. And in high grades, E67 gets expressed in the G2M or in this aneuploid population over here. Very important because this is almost allowing us to stage where you are in cervical precancer using our cytology specimen that we're looking at. This is some data actually from China where we look at proliferation post G0, G1. We look at E67 overexpression, and we're looking at single cells up here, squamous cells over here. We have 3 papers describing just by light scatter what squamous cells look like versus columnar endocervical cells look like. And again, we can look at proliferation, E67 overexpression in a L-cell patient. What has that yielded? Well, if you look at all the different companies doing HPV DNA, this is a study we did on over 3,000 patients with over 600 biopsies. Roche, Genpro, Hologic, QIAGEN, the usual suspects. Here's NPV versus the Oncotech made by us. We have very similar negative predictive values, but the difference in specificity is profound. And this population right here is the number of women who are going to colposcopy who don't need it. That's what we're trying to avoid in the United States. A great comment was made by Phil Castle, a leading thought leader in HPV, who said, finally there must be a greater appreciation that both clinical sensitivity and specificity, or their complements, false negative and false positive results, are important to patient safety. What we heard from Digene and now from Roche, all you ever hear about is negative predictive value and sensitivity. They're pretty much the same thing, right? You never heard Digene ever talk about positive predictive value and specificity because it was poor, okay? In fact, it's analogous to HIV. The first HIV ELISA had a 99% negative predictive value and a 20% specificity and positive predictive value. And we developed it out. We would never report out an HIV ELISA unless we did a Western blot, right? To give us the specificity that we needed. Doing HPV DNA alone, there's no Western blot. The HPV DNA has almost the identical performance characteristics as the HIV ELISA. High negative predictive value, poor specificity, and poor positive predictive value. You can also do this in head and neck. Combination on it. Here's the HPV DNA positive sample, E67 positive cells here, PD-L1 expression here. What was really interesting is that the E67, the transformed cells, weren't expressing PD-L1. That may be a reason why in HPV head and neck cancer cases that these are actually not ignored by the immune system and might be cleared. Here's another case, E67 overexpression, PD-L1 positivity, no PD-L1 expression on the E67 positive cells. So those are able to be scavenged by the immune system. And of course, here's HPV negative case. So we use the same strategy with lung cancer, where we use a non-cross-linking fixative. We are able to make a single-cell suspension with this incellPrep device that I showed you. We take a small punch, we make a single-cell suspension, and we're able to quantify PD-L1. So what we're able to do, because we're using fresh tissue, is we can finally quantify PD-L1. In fact, we can run MESF beads with the flow cytometry, and I can tell you exactly how many receptors of PD-L1 are on a cell-by-cell basis. And we're also able to quantify tumor-infiltrating lymphocytes, which have important prognostic information, and obviously we're able to quantify aneuploidy. And again, this is a control cell cocktail that we've lyophilized and made. It consists of PD-L1 positive cells, PD-L1 negative cells, and PBMCs all in the same control cocktail. And you can see this very nice CD3, CD8 curve. We can do a CD4, CD8 ratio in this control cell cycle. Obviously, measure aneuploidy and PD-L1 expression on the epithelial component. These are control cells. This is a tumor. They look almost identical. It's beautiful that we've been able to create a control cell that looks almost like tumor. There's a diploid lung cancer. We can quantify CTLs. It's diploid, very little PD-L1 expression. Here's an aneuploid lung tumor. Again, we see CTLs, it's aneuploid, and we see PD-L1 expression. If you've ever been to a pathology meeting and they talk about PD-L1, you go up and introduce yourself and you say, what's your clone? Right? So I'm sure all of you have heard you know, with PD-L1, hi, what clone are you using? Or if you talk to the pharmaceutical companies, what's your clone? 28A, you know, the other ones that I don't use. And then for Insight Pharma, we used all 4 clones at the same time by flow cytometry. And you know what? They're equivalent. They're almost identical, except for the clone that's intracellular, and then there's clones that are cell surface. There may be a difference there, but for the most part, they're the same. But they have different reactivities in FFPE. That just illustrates the struggles that cancer will. If you go to ASCO or AACR and they're talking about immune checkpoint inhibitors and PD-L1 quantification by IHC, it's the biggest discussion on the world, and it's been, frankly, a disaster. One of the big problems is histiocytes staying for PD-L1. So you see a big cluster of brown, you don't know if it's a cluster of tumor or a cluster of histiocytes. That's what our pathologists at Stanford always struggled with. And so you don't really know what the quantification is. That's another reason why creating a single cell suspension and doing very fine quantification has distinct advantages. We're also able to look at the immune cell profile, CTLs, helper cells, and MACs, look at differences in the different profiles in non-small cell lung cancer versus normal. These obviously have prognostic significance. Proliferation post-G01 also has prognostic significance. So there's just so much more we can do when we create a single cell suspension. These are the survival curves in some of the studies that we did, looking at CD8 TIL quantification. Obviously, if you have higher TILs, you have higher survival, less TILs, lower survival across the board. This is another interesting look and where we did DNA content versus PD-L1 expression. And again, you see these incredible aneuploid populations expressing tons of PD-L1, where the diploid tumor was not expressing PD-L1 at all. And you can see, you can actually count the number of tumor cell clones, 1, 2, 3, 4, 5, 6, 7, different populations of tumor cells within the tumor that you have to account for because they're expressing different levels of different markers. And when we looked at this PD-L1 expression versus heterogeneity index, which is essentially the number of tumor cell clones, sure enough, as the amount of DNA increases on a per cell basis, PD-L1 expression goes up. That's tumor mutational burden, except this costs us about $80 instead of $5,000. Okay, so these are some other things we're trying to bring to the world is we need to be much more cost effective than charging a patient $5,000 to do a tumor mutational burden to see what the PD-L1 expression should be because the IHC was so bad that they couldn't tell in the first place, right? That's where we are in pathology, is that some tests are inhibiting use of drugs. I used to teach a case in Stanford too, where there was quantification of CCR5. It was a big, CCR5 inhibitors were a big potential drug in HIV. They blocked the receptor that HIV bound to. The problem is the test that was used to say you qualify for the drug was $3,000, and it wasn't covered by insurance. So that was the first case of how a diagnostic killed a drug market. The drug companies projected sales in the $1 to $3 billion range. The drug sold $50 million at its peak because the diagnostic test was so expensive and nobody could pay for it. And also we got to get away from this CLIA lab model that we have in the United States where to get your tumor mutational burden, you have to send your sample from Lebanon or South America or Africa or someplace to Foundation Medicine in North Carolina and get charged $5,000 on top of it. That's a big problem. So the other big issue is being able to disseminate technologies. incellDx is a kit company. We sell our kits to Benta. Benta sells our kits in the Middle East. It's an easy model. It brings these diagnostics to to your hands, okay? You don't have to take your clinical samples and send them across the pond and get charged an exorbitant amount of money. So again, PD-L1 and DNA content, this will come up again. This is microfluidics over here. This is off a CTC chip. And we'll talk about CTCs, which we're also bringing to Lebanon in the Middle East as well. But we're able to do DNA content on CTCs, which no other company can do. And so there's a great example in prostate. If you look at benign prostatic hyperplasia, they have circulating epithelial cells, but they're benign. I don't even call them CT, I call them CECs, circulating epithelial cells, right? Because we don't know they're tumor cells. In fact, in BPH, they're not tumor cells. How do we know? DNA content and ARV7. Yeah. But fact is, we don't know all the time what's circulating in blood. Is it low grade? Is it normal? Is it tumor? That's also a big issue in CTCs, is being able to characterize those cells without having to pick them, isolate them, sequence them. There's a story from these colleagues of mine who make the CTC machines that one institution, I think it was Mass General, did an experiment where they were looking at a particular tumor, very heterogeneous. They did CTCs, they picked the cell, they sequenced for 1,000 genes. You know what it cost to work up that patient? $50,000. Now that sounds like a great clinical assay, right? $50,000 to work up that particular patient's tumor. We don't need 1,000 markers on cells. Because if you keep the cell intact, if you do 100 markers by NGS, you give the bioinformatics guys 100 parameters. If you do 20 markers on an intact cell by flow cytometry, you get 20 factorial combinations of markers on that particular cell. That's 10 to the 18th power parameters for the bioinformatics. That's the importance of keeping the cell intact. That's the importance of cytology and looking at morphology. When I was going through pathology, I loved cytology. I was telling the gentleman earlier that I happen to be really bad at surgical pathology. I don't know what it was, like my eyes didn't work, or I'm a terrible artist, so I had a bad spatial relationship. Whatever it was, I was horrible at surgical pathology. I was good at cytology. And cytology, you know, was a different bird. And now we're able to measure an enormous number of markers on that single cell, including morphology, and incorporate those into treatment models and in prognostic models. So we did a study in lung cancer, and you've probably heard of all these markers: LAG3, TIM3, CTLA4, PD1, PDL1, TIM3, and markers for CD103 is my favorite. It's a marker on T cells. It says it's directly engaged with tumor. So it's an important marker by flow cytometry. And we did them all at the same time, all on the same cells. We actually did a cancer marker panel of 12 markers as well. But the bottom line is we were able to do 20 markers at the same time. And also do CTCs with a subpopulation of those markers, and use a system where we're able to reproducibly get 5 cells out of 10 million. So 1 in 2 million CTCs we can detect reproducibly. The two things that you've never seen in any journal yet, right, are reproducibility of CTCs, which I'm showing you right here is in our latest paper. And has anyone ever seen a serial cell-free DNA on somebody with tumor? Like you draw blood day 1, day 2, day 3, day 4, day 5, and look at the concordance of those blood draws for cell-free DNA or cell-free RNA. I was talking to a Nobel laureate friend of mine, Andy Fire at Stanford, about a month ago, and I said, Andy, have you ever seen cell-free DNA over a time in the same patient? Or, you know, and he goes, and he said, no. I said, I haven't either. You know what he said to me? You never will. you won't see it, right? Cell-free DNA is great when you find something, but when you don't, you have no idea where you are. And will you find it 3 days from now? I have no idea, right? Because it's imperfect. Now, pregnant women is a different story. They're spitting, fetal red cells into the peripheral blood. Yeah, I get cell-free DNA for prenatal diagnosis, but not for solid tumors. So we found an interesting thing when we were able to do this 20-color panel plus cell cycle. We found that there was very distinct profiles in the diploid tumor versus the aneuploid tumor. So for instance, in the diploid tumor, we saw EGFR expression and no ALK fusion. In the aneuploid tumor, we saw EGFR and ALK fusion. The other thing we found, as I'll show you in a minute, aneuploid tumor cells end up in the periphery. That's not a big surprise. They're proliferating like crazy. Their DNA is completely messed up, and we need to be able to treat that cell that is going to end up in the periphery and end up metastasizing. The other thing was we did PD-L1 quantification in the tumor. So this was an aneuploid PD-L1 tumor with lots of CTLs with 243 CTCs expressing PD-L1. Okay? This patient got PD-L1 therapy. Here's another patient. Aneuploid, so bad actor, 0.6% cells with PD-L1 expression, which was the lower limit detection of our flow cytometer. It was called negative by IHC for PD-L1. But you know what? There's PD-L1 positive CTCs. This patient should have gotten drug and didn't. So the way we're looking at things from a pathologic standpoint is we're giving this overall, all-encompassing look at tumor cell biology, both in the primary tumor and in blood through CTCs, so we can have a comprehensive strategy for treating those patients with a focus really on treating what's going to spread, right? You always hear the adage, you don't die of the primary tumor, you die of the metastasis, right? Well, hell, let's treat the metastasis. Or better yet, make sure we treat the metastasis, okay? Because they could be different than the primary tumor. We certainly want to treat the primary tumor, but we also want to make sure that we're treating the cells that are circulating. Here's just some images, and they're cool to look at. Here's PD-L1, CTCs, white blood cells captured with us. Here's what's maybe what's called a camel, which is the metastatic theory where there's a fusion of macrophages and tumor cells. That is the putative metastatic cell that has both characteristics of a macrophage and characteristics of the tumor cells. They happen to be CCR5 positive. So now the CCR5 inhibitors that we used to use for HIV are now being used in cancer to keep those metastatic cells from migrating into tissues. That's where we are in the biological world these days. So what we did is, with our 10 to the 18th power parameters, we found this set of parameters that defined who had CTCs up here and who didn't have CTCs down here. And that was highly statistically significant. And look at this, it all depended on the TILs. Very few of the tumor markers had significance, but the TIL markers did. We actually found that it's the CD4 T cells that were most associated with circulating tumor cells, not the CD8 T cells, which is very interesting. We found the cell type, like I said, CD4 positive, CD103 positive, LAG3 positive, CCR5 positive, which is a T-reg cell that if they were there, you had a CTL. You had a CTC. So we're starting to be able to predict from the primary tumor who's going to have circulating tumor cells and metastases and be able to treat those at the same time as we're treating the primary tumor. Bladder cancer, which will be the last topic I'll talk about. We just developed an assay from the study that we did with Henry Ford. We looked at 3 different fixatives, the horrible alcohol-based fixatives. That's my second beef besides the cross-linking fixatives. and our incellMax for recovery of cells from urine. In urine, everyone's going for cell-free DNA, cell-free RNA, and exosomes in urine to try and predict tumors. Why? Because the recovery of cells from urine is horrible, especially when you fix in the alcohol-based fixes, preserves, and liquid prep. When we look at inadequacy rates by flow cytometry, single nucleate cell recovery from urine, far greater in incellMax than the the alcohol-based fixatives. If you look at epithelial cell recovery, again, the same phenomenon. We lose so many cells in these alcohol-based fixes. Why? Because frankly, urine is hypotonic. It makes the cells swell, it makes the cells fragile. It's very hard to recover cells from the urine. We use incellMax, which is isotonic to the cells, and it preserves them. And it's been great for bladder cell analysis. This is the float on normal urine. You can see the importance of doing aneuploidy and DNA content on bladder cancer samples. You see all these aneuploid peaks in these tumor samples. PD-L1 expression in tumors. Look at the ROC curves. All of these with AUCs around 0.9, 0.85 for these particular markers. What were the markers? Percent aneuploid epithelial cells. The DNA index, percent single nucleated white blood cells, and PD-L1 expression on white blood cells. Not PD-1, PD-L1. This was a really important parameter that almost defined bladder cancer in of itself. We don't know why, but we know it's a real big driver in our bioinformatics models. So when we looked at our bladder cancer study in over 50 patients, look at the statistical significance of PD-L1 expression on white blood cells compared to normal. Bladder, normal, cancer, normal, cancer, normal, cancer, normal. Percent white blood cells, also statistically significant. This is essentially proliferation, also statistically significant. DNA index, statistically significant. What wasn't statistically significant, ironically, was a PD-L1 expression on the epithelial cells, which we wouldn't expect anyway, knowing the variability of PD-L1 on tumor cells. These were statistically significant. We used a genetic algorithm to select the features to put into the algorithm. And to cut to the chase, we were able to develop a malignancy score for bladder cancer out of urine by flow cytometry that generated a sensitivity of 98% and a specificity of 87% in patients with microscopic hematuria, of which there's about 1.5 million in the United States every year who come into their general practitioners or urologists, and nobody really knows what to do with them except give them cystoscopy, right? I don't know anybody who wants to have a cystoscopy. So we developed this test to be able to screen patients to decide who really needs cystoscopy and who needs closer follow-up and repeat testing in 6 months. And we just launched this in the United States. And look at the continuum in microscopic hematuria in terms of cancer. It's all over the place. So we're now trying to decipher who's low grade and who's high grade to give clinicians some guidance as to what they're looking for when they do go to cystoscopy. And so I won't go into it anymore. Again, we use the same type of model in breast. Find needle aspiration, core biopsies, make a single cell suspension, put it in our particular fixative, allows us to do HER2 antibody, HER2 mRNA, ERPR, CD45, MCV, NTDH, which is a marker that we found in metastatic breast cancer, ECAT here, and CD44, which is a tumor stem cell marker, and we're able to do bioinformatics and separate cancer from normals simply on a fine needle aspiration or core biopsy. And you know what these 3 cases were?
I think because of the hyperplasia.
DCIS. Yeah. So Genomic Health will charge you $5,000 to do something similar to this. We're working on this right now with MD Anderson, and hopefully someday this will become our next great diagnostic, and we'll bring it over here. So in summary, what we try to do is create these test bundles and offerings that completely differentiate in the market. For instance, like HPV, we want to be a lot more than just saying you're infected and what's the risk of being infected. We want to tell you where you are on this continuum of cervical cancer, give the clinicians more information to go on. That's our job as pathologists. And you know what, frankly, the word that I really don't like is risk. So if you have BRCA, you have this risk of having breast cancer, this risk of having ovarian cancer. I wrote an article that said, when you talk about risks with a patient, what you're actually doing is putting the onus on the patient to decide what they want to do. What a horrible decision for women and men sometimes, that you say, you know what, you have a risk of breast cancer, ovarian cancer. Do you want to do a mastectomy? Do you want to take out an ovary? Do you want to take out your prostate because you're at high risk? Can you imagine that decision? I think it's a failure of us in diagnostics that we're at a place where some of us, not usually pathologists, but these companies who are making these tests, come out with risk, or say you have a 50% risk or a 70% risk. Back in the day, that meant you have a 50% sensitivity for cancer, or 50% sensitivity for a disease, and it was a bad test and you just threw it away, right? Well, now over the years, what I've seen evolve over 30 years of pathology is they're now using that to define risk. You're at increased risk. And then they tell the patient, you're at increased risk of this, that, and the other thing. What do you want to do about it? And then the clinician try and guide them, but none of the choices are good. So I think we have, as I was saying before this talk, I think as pathologists we have to come together. We have to lead the way, not the others, the big billion-dollar companies. We need to lead the way in saying, this is what we need to do to make better decisions, to give you clinicians more information to manage patients, and this is how we can do it. And like I was saying before, I think we can help solve some of these conundrums, like how do we get good nucleic acids out of FFPE blocks. I remember at Stanford, our surgical pathologists used to have fits when they would get a block back from molecular pathology and it looked like Swiss cheese, because they were taking punch biopsies out of this FFPE block. Or they were taking, 80 micron sections, this, what we used to call beefsteaks, right? And then doing that in nucleic acid extraction, and you came back and you had 5 microns of a tissue block left by the time molecular pathologists were done with it, right? You gotta find a better way. One of them is to, I think, is to do what we do for the lymphoma protocol. We take a small piece, maybe we freeze some for the molecular biologist, maybe we just take some and make a single cell suspension, and we can do everything out of that. And then we put the rest in formalin and do the morphology. And maybe we think about ways to use other fixatives that may not look the same as formalin in some cases. Maybe we can be trained to see how that may look, but at the end of the day, they allow us to do a lot more out of that tissue block. But anyway, it's been a pleasure speaking to you. I love talking to pathologists. I feel like it's family. We can air our dirty laundry. We can talk about what everybody's trying to do. And but I do think, just like any family, we can come up with very meaningful solutions. And we can drive what people use, and we can educate the clinicians, the oncologists, and our constituents, educate them on how to use tests. Because, when you talk about test statistics and how to use test performance, you'll get blank faces from a lot of clinicians. Obviously, you won't get it at MD Anderson, but the fact is, it's our role to educate the clinicians on what this information means. And it's a very serious role, and we should not take it lightly. And it's been a pleasure talking today. Thank you.
We have roughly 10 minutes for questions or comments or discussion. Please.
We already are doing the HPV. We started with the HPV. What also they are coming?
Right now, HPV, lung, prostate, bladder, the launching is this week.
Breast not yet.
Breast is coming to the market also. Head and neck is present.
Yeah.
Head and neck E6, E7 with proliferation, or head and neck with PD-L1, E6, E7 and proliferation.
Are both available here now.
Yeah.
So in one test, and it takes, it's a one.
Yes, in Lebanon.
Where?
In which laboratory?
Yeah, that's
At Benta. Benta and other laboratories that we are working with. Yeah.
And I think breast, the first iteration of breast will be just a very simple HER2 antibody, HER2 fish, ER/PR, and ploidy by flow cytometry. So again, you'll have a one-day turnaround time. You won't be in this dilemma of doing IHC, and if it's equivocal or 1+2+, you reflex to fish, then you wait another week or 10 days to get the fish results. You'll get it all, and the assay takes 4 hours. I can envision, a one-day, two-day
And who
turnaround.
interprets the results? We give you the sample and you send us the results, or it is for the pathologist to interpret them?
Right, so you'll get HER2,
I'll get the results.
you'll get the results.
And cost-wise? No idea.
Cost isn't.
That's not really-
Put it this way, it's my philosophy that I said in the beginning. We're not in the business of creating $3,000 and $5,000 tests. We want to create tests that are just what we would usually expect from multiple markers. In the United States, we have CPT codes. So for instance, our bladder test reimbursed at $400. Our HPV test reimbursed at $250, right? I'm not saying that's what it will cost here, but I'm saying we work backwards.
This range.
Yeah, that range. We work backwards in the States to try and figure out what would be, the best possible pricing.
There is anything on lymphoma protocols?
Do I actually have it?
Yeah.
I have it. The CAP protocol for lymphomas that says take a small piece of tissue, push it through a mesh, do your flow cytometry.
I'll give you all the results.
What's that?
It will give you all the results after that.
That's right. So, that was always my rebuttal. I mean, some people said, well, you're not doing the standard of care. I'm like, do you do lymphomas? Yes, I do lymphomas. Do you take a small piece of tumor before you put in formalin and make a single cell suspension for flow? Yes, we do. I said, so I'm proposing for lung cancer. Do the same thing. Do it for all solid tumors, because we have this FDA-approved device to do it, right? So, might as well.
For bladder cancer,
Yes.
if you have the PD-L1 negative tumor, what will be the interpretation of your test? Is
Well,
it
again-
FDA approved, or
So we have this.
to delay the cytoscopy? The cystoscopy?
So you're talking about two different things. There's two different results that we get from our bladder cancer test. One, high risk of bladder cancer.
In
So
your-
one is it bladder cancer or not bladder cancer?
Okay.
The second one is on the tumor cells, are they PD-L1 positive?
Yes, no,
So
I'm
you
talking
could-
on urine, on the screening for.
That's what I'm saying. You could conceivably have a result that says this is bladder cancer or high risk of bladder cancer, but it's PD-L1 negative.
Yes.
So you'd have to go to a different treatment option than immunotherapy, right?
You must have a cystoscopy to see if you need to have a low-grade tumor, for example. You can
Right.
have a
Oh no,
low-grade
we're not trying to eliminate cystoscopy.
Okay.
So if we get a result that said bladder cancer but it's PD-L1 negative, of course you go right to cystoscopy.
So what is the benefit of, okay, so you know that you are
Right.
dealing with a
The
high-grade
benefit is
tumor.
only 3 to 5% of people with microscopic hematuria
Okay.
actually have cancer.
Okay.
So that's our screening.
And you are dealing with high-grade tumors that are aggressive.
Well, in that population, depending on age and smoking history, we're dealing with a whole spectrum of disease from low grade to high grade. So we took all comers for that intended use. We didn't split them into low grade and high grade. Okay, we just took all bladder cancer. Now, we're gonna break the code and see who was low grade. Like I showed, there was a continuum. And I suspect that continuum is being driven by the fact there's a fair number of low grades and fewer high grades, which were far away from where the normal cells were. So I think there's really a continuum in the bladder cancer spectrum. But there's no doubt we're picking up the low grades.
Okay. But it's interesting in not in screening essentially, but more in the surveillance of recidive or
Post-therapy.
post-therapy.
Yeah, that's our next study.
Okay.
Perfect.
Yeah, that was a little bit one of the questions I wanted to ask. incellDx tests, there are packages, like 1, 2, 3, 4, 5, right? Those
Correct.
ones.
Yep.
And approximately how many countries are they
So
working today?
We have,
Roughly.
well, we have China, which I could retire just doing China.
That's 10 countries.
Right? Yeah, right, exactly.
Okay.
We have,
Continent.
yeah, US, China, Central America, South America, Europe, and Bent is doing our Middle East.
Okay, and the question is, any of those tests useful for screening normal people for cancer?
Yeah, the bladder would be.
The bladder.
Oh, not, yeah, for microscopic hematuria, not normal.
That's the only one that would be for screening.
Yeah, most of them are reflex or to glean more information.
GYN1 would not be as a substitute for vaccine.
No, it could be, the best use of it is in conjunction with pap
Yeah,
smears.
sure.
So in a co-testing model.
But for the suspicious ones, not for everybody. Because we screen for pap smear everyone. We cannot use it to screen everybody.
Exactly, but
Just
suspicious
suspicious
ones, so
ones.
ascus and above,
Yeah.
yeah.
Thank you.
Yeah, out of the same sample. You'd have to re-swab everybody.
Thank you, everybody,
Thank you.
for coming. Thank you, Bruce.
Thank you. Thank you.